And now I will continue with the intervention of Walter Longo. Walter Longo, fasting, it's his job. L'intervention de Walter Longo, l'apôtre, j'irais dire, du jeûne thérapeutique. Merci les amis.
So we started a long time ago trying to understand the fundamental connection between food and aging. That was my interest, actually. And then I started, maybe 15 years ago, I started to think about this connection, something that we knew for a long time, which was if you starve yeast, and this is a unicellular eukaryote, a very simple organism, if you starve it, as you see at the bottom, the yeast becomes much more resistant to all kinds of toxins, what we call multi-stress resistance. And so this resistance actually can go into the 50-fold, 100-fold increase, right? So they're much, much more resistant. And you see that without going into the details, the starved yeast, each dot signifies each position signifies a 10-fold increase in resistance. And so then we did a lot of work on the connection between food, ingredients, components of the food, for example, protein, amino acids, and the genes and proteins in first in the yeast organism, and then eventually in mice and humans. And so we figured out that, in fact, proteins and amino acids control two of the major pro-aging pathways. One is called growth hormone IGF-1, and the other one is called TOR-cis kinase, and these are now very well confirmed to be very essential in the aging process. And then my lab, a long time ago, also discovered the IGF-1 role in aging was discovered by Cynthia Kenyon and Gary Ruvkun. And we discovered the TOR-cis kinase, and then we also discovered what we call the sugar signaling pathway, right? RAS, PKA, controlling, and both of them. So if you have a lot of proteins, if you have a lot of sugar, all these pathways are activated, and they make the cell grow, but at the same time makes all kinds of cells in the human body grow, but at the same time they make them weaker, right? So they make them weaker and age more quickly. And so I started thinking 15 years ago, this is really a great opportunity. Why? Because a lot of these genes that I'm showing you, these proteins that I'm showing you, are oncogenes or oncoproteins or proto-oncogenes, meaning they're the normal form of what gets mutated and it becomes a cancer-causing or cancer-promoting gene, right? So then it occurred to me that by starving cells, normal cells will all respond to the starvation signal by shutting down this pathway. So you remove the protein, you remove the sugar, these pathways are downregulated in normal cells. But by definition, the cancer cells, because they have these oncogenes, let's say in the RAS gene or even in the IGF-1 receptor, they're always stuck in the on mode. So this was really a great opportunity to distinguish all normal cells from all cancer cells. And so you starve, and we knew then later on we figured out, in fact, by starving a mouse in this case, but also something very similar happens for people, the IGF-1 is lowered by 70%, so one of these key signaling pathways is really shut down, also because the inhibitor of IGF-1 signaling called IGF-BP1 is up 10 to 20-fold, right? So you have IGF-1 being lowered, IGF-BP1, its inhibitor being going much higher, and also glucose being lowered. I mean, in mice, there is a drastic reduction of glucose. In humans, not as drastic, but there is a major decrease in glucose in people that are fasting. So then we started with simple experiments, both in the US and in Italy, which was, well, we have done a lot of work in yeast, which I'm not going to show to you. But the simple experiment was just if you take a mouse and you starve it, can you protect it by, in fact, lowering IGF-1 and lowering TOR signaling, et cetera, and lowering glucose? And the answer is yes. In fact, you see a major protection. This is protection against cisplatin and doxorubicin chemotherapy drugs. And on the left, you see the red line here are mice that were starved before for 48 to 60 hours before giving cisplatin. And here are the same thing, red line, and the gray line is instead the mice that received the normal food before chemotherapy. And so you don't see this very often. For example, what you see there with doxorubicin, they're all alive, all dead, based on whether they were starved or they were fed normally. Now let's see if this works. Okay, so then on the left, the mice that were starved before chemotherapy, and on the right are the ones that ate normally, and so you see that the mice starved before chemo, they're moving around like they didn't receive chemo at all, and the ones on the right, actually about 65% of them will be dead in 10 days or so. There is a drastic difference. And so, and I'll show you some clinical data in a second. So when we first went to oncologists over 10 years ago with this, and actually David Quinn is here, he was one of the first guys that we bothered, and they said, well, show us that the fasting and the starvation does not protect the cancer cells, because even a small protection of the cancer cells will have rendered this whole thing useless. And so we went back to the lab, and there was a reason, actually, an evolutionary reason for why this shouldn't happen. And the idea is that every mutation in evolutionary biology, we know that every mutation, most mutations are deleterious, right? So cancer, because there's so many mutations, at least the hypothesis was that when you put the cancer cell in a complex environment like starvation, you really change everything, and everything is different from what it was when the cancer evolved, right? So the cancer forms slowly, and in that time, there are a lot of proteins, there are a lot of sugar, there are a lot of carbohydrates, and so the cancer now finds itself in a very different environment, and the idea, or at least the hypothesis, is that it will struggle in this environment. So starvation, so the best thing you can do for cancer cells is to keep giving the cancer whatever, the maximum nutrition, right? So the cancer benefits much more from a lot of food than the normal cells, because the normal cells have, as I just showed you, a very, they're able to switch to a starvation response mode. Anyway, so then we started to look at this. It is, in fact, a cancer cell confused in this environment. And I'm not showing you those data. Well, actually, yeah, no, there is not the fasting alone. But already fasting alone in most of the experiments that we did, so these are cycles of only about 3 days of fasting every couple of weeks or so, 1 to 2 weeks. And over we showed that in most cases, the fasting cycles alone were as good as chemotherapy. So the cancer progression was delayed as much by just giving the short cycles of fasting as it was by giving high-level chemotherapy. But I think the important thing is, and I'll show you some slides later, is that the combination seems to be much, much more powerful. Now you have a synergism, right? If you just do one or the other, as most people try to do, we always try to go the natural way or the pharmacological way, and there's two groups that really don't like to talk to each other. But really, when you combine the fasting, as you see there, with breast cancer, and this is just one cycle of fasting plus chemotherapy. In the red, you see that if the mice are fasted and given chemotherapy, now you start seeing long-term survival, and this is neuroblastoma on the right. Now you have 50% of the mice that are cancer-free long-term, but only in the combination of fasting and chemotherapy. And this is actually a study, and I just want to show you, focus on the two panels on cancer-free survival here. This is a study done by a group in Switzerland looking at lung carcinoma and mesothelioma. And in here, you see the control. This is cancer-free survival. So what's the percentage of mice that are cured? So of course, if you don't treat them, 0 are cured. If you give them chemotherapy, 0% are cured. If you give them fasting, 0%. And then you combine fasting with chemotherapy. And you see it goes up to 40%, and here actually 60%. And it's very similar results now we see over and over in many different tumors. So the idea is that, as shown here, normal cells, the normal organism, you starve them. There is a very coordinated response. Why? Because we have always been exposed to starvation periods, as Jean-Jacques told you earlier. Historically, we were in a very different world, and starvation was very, very common. And it is very, very common for any organism that you can think of. So normal cells are very well adapted to this. There's lowering of IGF-1, lowering of glucose. They know what to do. They increase the stress-resistant transcription factor. One of them is called FOXO. And they go into a protected, highly protected mode, anti-aging, and also anti-carcinogens. Tumor cells, instead, everything happens the same outside of the cell, but then any oncogenic mutation, which of course all cancers have, is going to lead to rebellion, essentially, the inability to respond. And these leads, of course, if you have low glucose, you have low IGF-1, and you refuse to acknowledge that this is the environment, then sensitization happens, and you struggle and you die. I always say it's like going to the desert with no water and no shade, and you're running, right? So this is what cancer cells do. As long as you have water and you have shade, you could probably run in the desert. But if you now remove the shade and the water, it's just a matter of time before you're going to die, right? So, and this is the idea that we're trying to bring into the clinic. Now, something else very quickly is what we've done recently with dexamethasone. Something that we backed the clinicians to do is to remove dexamethasone for patients that didn't need it necessarily, so for whom it wasn't absolutely required, because we knew that dexamethasone increases glucose levels in the blood. And also we knew that there were data showing that the patients that have higher glucose levels respond worse and do worse during the therapy. So you see there both the use of rapamycin at the bottom and dexamethasone, which is widely used before chemotherapy. And in both cases, you see this effect on glucose, so hyperglycemia, and the same happens in patients. And there, what we just published is look at the effect of these widely used drugs and the resistance to chemotherapy. So this is chemotherapy alone, survival after chemotherapy alone, and look at survival after chemotherapy plus dexamethasone. So these are, at least in the mouse, has a devastating effect in making the chemotherapy much more toxic to the normal cells, particularly to the cardiomyocytes in the case of doxorubicin. And here is the same for rapamycin. And then here we show that you can, just by using insulin, so this is the sensitization caused by dexamethasone, and this is just the use of insulin together with the chemotherapy. But look at what happens when you use fasting together with chemotherapy. Now you completely reverse these sensitization effects of the dexamethasone, and same thing happens for rapamycin. Just again, the importance of mechanisms, the importance of understanding how things work, because then you can go back to the clinic with a much, much clearer idea of what to do. Now, we still haven't convinced the clinicians to remove dexamethasone. They tell us, so, in many cases it's necessary, but I think, I would be very worried if I were a clinician giving dexamethasone the day before giving chemo. Okay, so then something that we recently published was what if this damage done to the cancer cells would make the cancer, make the immune system more effective in attacking the cancer. And Guido Kromer had done a number of papers looking at chemotherapy and the possibility that the chemotherapy would predispose cells to the attack by the immune system. And so we looked at this and moved to fasting mimicking diets, because most cancer patients don't want to fast, and show that here the combination of this fasting mimicking diet, this is a low protein, low carbohydrate, high fat ketogenic-like diet, but it's also calorie restricted, severely calorie restricted. So you see here the infiltration of T cells, of immune cells in the tumor is not very high if you give them nothing. It's not very high with the diet. It's not very high with the chemo. And now you combine the chemo and the diet, and it goes much higher. And then we actually, I'm not going to talk about the details here and the mechanisms, but certainly just to show you that by knocking down by knocking down the immune system, by inhibiting the immune system, now the FMD, the periodic fasting mimicking diet is no longer effective, meaning that it's acting, at least in this breast cancer mouse model, is acting by activating, the FMD is slowing down cancer progression by activating the T-cell dependent attack of cancer cells. And this is just a mechanism where then we figured out some of the genes that are involved. This is heme oxygenase 1, which is a mitochondrial enzyme that breaks down heme. It seems to be the lowering of this heme oxygenase seems to be responsible for the sensitization effect and for the opening up the cancer cell to the immune system. Okay, so this is the paper the Guido published at the same time with ours. And I don't know if he's going to talk about it, but Guido had focused on cancer mimicking, and fasting mimicking drugs instead of fasting mimicking diets. Okay, so now in the last 3 or 4 years, we've been focusing on what's next. So the starvation and the fasting mimicking diet now are causing this very wide effect. And now in collaboration with Alessio Nencioni, we now start combining it with kinase inhibitors. And you see here in a lot of scenarios with lung cancer and breast cancer and colorectal cancer, now you can combine this kinase inhibitor with the cycle of the fasting mimicking diet. And in each case, you have additive and sometimes synergistic effects. Now we're also doing it with hormone therapy. It's working very well, and all kinds of other combinations. Okay, what about clinical data? Still early. This was something that we published a long time ago. It was a case study, case report of 10 patients, 6 of whom had done the same chemo, either with or without fasting, and the side effects appear to be lower, or at least they report lower side effects, going from fatigue, weakness, headaches, nausea, vomiting, etc., when they fasted together with the chemotherapy. These were cancer patients that had all kinds of cancer, so very preliminary, but very promising. Then the study that David Quinn, Tanya Dorf, and myself finished. It took us a while because patients don't like to fast, but if it was finally finished and published, and again, this is a feasibility study, but showing that patients that were on 72 hours of fasting together with the chemotherapy seems to do better, or at least reported doing better, a lot better than patient that did 24 hours of fasting. This is a nice control because it tells you that against the 24 hours, it seems to be superior, of course, very small. But this is another study by University of Leiden, looking at, this is a randomized but very small study, looking at the effect of combination of fasting and chemotherapy on erythrocyte level and thrombocyte level, and again showing that the patient that fasted together with chemotherapy were protected. I mean, the blood cells were protected against the chemotherapy toxicity. This was also shown at the level of DNA. Now I hope that the work from Charité will be shown later today. And this was on 34 women, randomized crossover trial, looking at fasting mimicking diet, a low-calorie fasting mimicking diet similar to the ones that we're testing. And I think this was worked out very well, and you're going to see it later, I hope. And then we are now close, but we're maybe midway through, a little bit better, with multiple trials, one on breast cancer, 60 patient randomized at USC, Mayo Clinic, and I think MD Anderson now joined. There is another one on prostate, and that's almost over. Hopefully soon we'll have the results. And there is another one on prostate cancer that is fasting mimicking diet, chemotherapy, but it's in much earlier stages. And the Leiden University said 250 patients randomized on breast cancer, fasting mimicking diet called Chemo-Leave, and breast cancer drugs. And that is about halfway through. So hopefully soon enough we'll know about the results from the midpoint. So just to summarize, I used this slide that we just recently published together with a group of oncologists at Istituto Nazionale Tumori in Milan. And the idea was that we're focusing with all this precision medicine, and that's great. And I think, as I've shown, it's very powerful. But I think that the fasting mimicking diet, and potentially some ketogenic diets create a much wider net affecting glucose, insulin, ketone bodies, IGF-1. And this is just few of the hundreds and hundreds of genes and factors that are affected. So the point that we made in this cancer discovery article was that the combination is really, we think, the future. So generate a hostile environment that has differential effects, hostile for the cancer cells, not hostile for the normal cells, and then at the same time use the targeted therapy to have a very powerful effect, hopefully in a precision manner against the cancer. And so together we may start seeing, as I just showed you earlier, for example, immunotherapy. Now we're combining with immunotherapy, and the hypothesis is, can it make, maybe sometimes the immunotherapy can work on 20, 30, 40% of the patients. Can this starvation, the fasting mimicking diets, expand this and make many more patients respond to these targeted therapies? And that's all I have. Thank you very much.
I would like to say I didn't have any chemotherapy, but diet, fasting is working too without chemotherapy. Thank you very much. Bye-bye.